So, from today we will discuss physical anthropology. As you know, in paper 1 and paper 2, for this particular batch we completed anthropological thought first, unit 1, 6, 7, 8. Then we went for paper 2, tribal India 6, 7, 8, 9. Then Indian society 1, 2, 3, 4, 5. Then we went for socio-culture anthropology 2, 3, 4, 5. So, 4 modules are completed: anthropological thought, tribal India, Indian society and socio-culture anthropology. Next is physical anthropology. So, what is this physical anthropology? Biological part is present in anthropology. First we have to understand what is the necessity to have a biological part in anthropology. The biological part will help us to understand evolutionary process. Anthropology is all about understanding the evolution of man biologically and culturally. So, up to now we understood culturally, like society, culture, marriage, family, kinship, economic systems, political systems, religion, then how the tribal societies are there, how the Indian villages are there, how villages will be there across the world. So, now we will be entering into the biology part. So, biology part means not again fully biology part, and helicopter view we will tell that. Helicopter view means on top view. So, when we see from the top, we will see only the basic structures we will see only skeletons, we will not see inside, we will not go deeper. So, we need to have a helicopter view of biology, not microscopic view of biology. We need to have a helicopter view of biology, but not microscopic view of biology. That is a major difference between a biology and a physical anthropology. Physical anthropology is different and biology is different. Biology is microscopic understanding of a man, whereas physical anthropology is a helicopter view of a man. So, what is helicopter view of the man means basic structures of the man, basic structures of existence. What are the factors which are responsible for the existence of the man? So, this existence, how it got impacted because of culture. This structures, how it got impacted because of way of living. How these structures got impacted because of some taboos, some rituals, some practices, some cultural things. So, how the biology of the man got impacted in the process of evolution, how it got impacted, we will be discussing in this particular physical anthropology. So, this physical anthropology mainly consists of paper 1 consists of 1.7, 9.1 to 9.8, 10, 11.1, 11.2, 11.3 and 12. This complete physical anthropology is 1.7, 9.1 to 9.8, 10 and 11.1 to 11.3. However, applications of anthropology, 12th is applications of anthropology, but we generalize into physical anthropology. 12th is applications. We will be applying the knowledge of anthropology into various disciplines, like in sports sector, like in nutrition sector, like in criminal identification, facial reconstruction, paternity diagnosis. If someone come and tell that he is my father or he is my mother, how we are going to have a paternity diagnosis, how we are using the knowledge of anthropology and we are understanding these concepts. These all are present in applications of anthropology. So, today we will discuss about 1.7. 1.7 per se does not have relevance in examination. There is no direct question in near past time. From 2016 or 2017, 2020, 2021, if you see the previous questions, there is no question from 1.7, but 1.7 is very important because of the fundamentals which are present in 1.7. It is optional or not. You have to know about the cell, you have to know about the DNA, you have to know about the chromosome, you have to know about the DNA replication, you have to know about protein synthesis, what is happening in our body. We have to know about cell divisions which are happening, which are sexual cell division, which are asexual cell division, which are mitosis, which are meiosis. So, in 1.7 what we cover, sir? In 1.7 we cover cell, chromosome, DNA, DNA structure, DNA replication, protein synthesis and cell divisions. These are all the topics which are present in 1.7. So, this constitutes physical anthropology, and exclusive 12th is called as applications of anthropology. And from 250 marks, physical anthropology carries 70 marks. Out of 250 marks, physical anthropology carries 250 marks. Again I am repeating, physical anthropology we have to not study from the biology point of view, we have to understand from the culture point of view. And when there is a question in paper 1, now only I am telling you to have a mental makeup. If there is any question in paper 1, socio-culture anthropology or evolution of man or archaeology or physical anthropology or applications of anthropology, socio-culture anthropology and anthropological thought, if there is a question, give priority to right answer from physical anthropology. Give priority to answer physical anthropology question. From first day of the class itself you make a mind. If in paper 1 comprehensive test, if there is a question on physical anthropology or socio-culture anthropology or anthropological thought or evolution of man or archaeology, the first my pen should go for physical anthropology. Why? Because it is science. When it is science, the fact oriented. When it is science, fact oriented, it will be correct or wrong, that is it. There will be no perspective, there is no interpretation, there is no subjective analysis. It is objective answer. If you write the answer, you will get the marks. But here also the examples, the cultural examples, the society's examples will give you one more extra mark. So, what constitute this 1.7? What constitute this 1.7? In 1.7, which is called as the biological basis of life, unit name is called as the biological basis of life, in which these are the things which are present in the syllabus. So, it is about the cell. It is about DNA structure and DNA replication. What is DNA? So, there is a structures of cell. So, before understanding the structure of cell, there are two types of organisms are there. One is called as eukaryotic, other is prokaryotic. Eukaryotic means the organisms where there is multiple cells. The organism made up of multiple cells is called as eukaryotic organism. Or the cells, if it is only single cell, unicellular organism, only one cell is present across the body, those are called as prokaryotic organisms. Eukaryotic organisms are we people, like human beings, we are having multiple cells. Whereas prokaryotic cells or prokaryotic organisms are those which are unicellular organisms which have only one cell, like bacteria, like blue-green algae, like amoeba. So, we are going to discuss about eukaryotic organisms. In eukaryotic organisms, there are multiple cells, like living beings, like human beings, like animals, like living creatures. Most of the living creatures on the earth are eukaryotic organisms, but not prokaryotic organisms. Prokaryotic organisms are few, like bacteria, like amoeba, like blue-green algae, etc. So, in this eukaryotic organism, cell is a building block. Cell is the foundation of this organism. So, what constitutes cell? So, numerous cells are there. How we have houses? So, every house have a border, every house have a compound. This is my compound, that is your compound, that is your compound. So, every cell is differentiated by other cell by a cell membrane. So, cell membrane is a closed circuit. This cell membrane will enclose all the cell elements into the cell membrane. So, what is present in the cell membrane? In cell membrane, inside the cell membrane of the cell, cytoplasm is present. A jelly-like substance which is called as cytoplasm is present inside the cell, and in that cytoplasm there is nucleus is present, and that nucleus consists of nucleolus. What is this nucleolus? Chromosomes. The chromosomes which are present in the nucleolus inside the nucleus of the cytoplasm constitute cell. So, cell membrane, cytoplasm, nucleus, nuclear membrane, nucleolus. Nucleolus is nothing but chromosomes. The chromosome, the pack of the chromosomes is called as nucleolus. And the cytoplasm means there is a nuclear membrane, there is a cell membrane. So, between the nuclear membrane and cell membrane there is cytoplasm. In this there is endoplasmic reticulum is there is ribosomes are there are mitochondria are there which are called as powerhouses of the cells. So, this complete constitute cell. Cell was first discovered by Robert Hooke in 1665 by observing slices of cork under a magnifying device, later named that slice as cells. These are the slices. These are individual slices which he observed. So, what is this slice? He told in the blood there are cells are there, numerous cells are there. Cell is the smallest structural and functional unit of an organism. It is a structural and a functional. It is a structure and every structure have a function. Structural and functional unit of an organism which consists of cytoplasm and a nucleus enclosed in a membrane, cell membrane. Cell make up of living things and carry out activities that keep organism alive. So, the cells, if they are living, the cells which are alive, the cells which are active, that is responsible for activeness of a human being. The existence of cell is responsible for existence of human beings. If cells die, the man dies. So, cells are responsible for the existence of man. Cells carry out certain functions, like there are several functions are there, like capturing and digesting food, excretes the waste, respires, reproduces. These functions helps to form tissues, in turn forms organs, eventually organisms. Cells combine to form tissues, tissues combine to form organelles, organelles combine to form organism, organelles or organs. Cells combine to form tissues, tissues combine to form organ, singular, organelles, plural, and organelles combine to form organism. So, what is the building block? The cell is the building block of an organism. The cell is the building block of an organism, and the cell is the basic foundation of human organism. The cell is the structural and functional part of a human organism. Cell is the smallest structural and functional unit of an organism. In this way, when the Robert Hooke has observed the blood, he started observing some slices of cork. This slices of cork he named it as. So, this is the cell. This is how the cells look like. So, this cell is separate from other cell, this cell is separate from other cell, this cell is separate from other cell. So, one cell is differentiated by other cell by what? A cell membrane. One cell is differentiated by other cell by what? By cell membrane. So, the cell, the parts of the cells, the components of the cells are enclosed within the cell membrane. So, this is cell membrane, this is cell membrane. What are present inside the cell? Mainly the nucleus is present. The main part is nucleus and the other part is cytoplasm. So, nucleus is the central part of the cell which is closed by the nuclear membrane, and the gap between the nuclear membrane and the cell membrane, the gap between the nuclear membrane and the cell membrane is cytoplasm, In this nucleus, nucleolus is present. Nucleolus is nothing but the combinations of chromosomes. The chromosomes together is nucleolus, which is enclosed within the nuclear membrane of the nucleus. And in cytoplasm we have mitochondria, in cytoplasm we have ribosomes, in cytoplasm we have endoplasmic reticulum. These are all the things which are present inside the cell between nuclear membrane and cell membrane. All things are not required, some are important like endoplasmic reticulum, ribosomes or mitochondria and nucleus, that we will be discussing. So, there is a cell membrane, there is a mitochondria, plasma membrane or cell membrane, cytoplasm, microtubules, lysosome or ribosomes, endoplasmic, these are endoplasmic reticulums, this is the cell. So, the components of cell we will discuss one by one. So, cells are of two types which are called as eukaryotes and prokaryotes. Eukaryotes are those organisms which have multiple cells and a nucleus, multiple cells eukaryotes. Example, human being. Prokaryotes are those organisms which have single cell, prokaryote, like bacteria, blue-green algae or amoeba, these are prokaryote. So, what are the components of cell? Cell membrane, which is also called as plasma membrane, cell membrane. Cell membrane, one cell is differentiated by other cell, one cell is different from other cell because of the cell membrane, because of cell membrane or plasma membrane. Protects the elements of cell and it separates, as well as what happens if sudden push is there, the cell membrane acts like a shock absorber, so that the shock will not go inside the nucleus and the nucleus will not be destroyed. So, cell membrane will act like a cushion. The cell membrane acts like a push factor. The cell membrane acts like a shock absorber. Protects the elements of cell from any external pressures. All the components of cell are enclosed within the cell membrane. So, first important component is cell membrane. The first important component is cell membrane. The cell components are enclosed within the cell, and this cell membrane acts like a shock absorber, which protects the components of the cell. If any external shock is there, external push is there, then the external push has been stopped by the cell membrane. Cytoplasm. So, inside the cell there is nucleus, and the nucleus is enclosed by the nuclear membrane, and the cell is enclosed by cell membrane, and the space between the nuclear membrane and cell membrane, the space between nuclear membrane and cell membrane, cell membrane, this particular space is filled by a jelly like substance which is called as cytoplasm. It is a jelly like substance present between cell membrane and nuclear membrane. Certain components like ribosomes, mitochondria, endoplasmic reticulum are present in cytoplasm. So, ribosomes, mitochondria, endoplasmic reticulums, these are the most important, like mitochondria, endoplasmic reticulum, ribosomes. Mitochondria is also called as powerhouse of the cell. The power is generated. The power is given to the cell for proper functioning from mitochondria. Mitochondria is also called as power of the cell because it converts food into energy. It converts the food. So, cell will get the food, that food is converted into energy, so that all the components of the cell will be active. So, all the components of the cell are active mainly because of mitochondria. Ribosomes. The major function of ribosomes is protein synthesis. If you see here, ribosomes are major components which is responsible for protein synthesis. Or what is protein synthesis? Blood is a protein, saliva is a protein, semen is a protein, hormone is a protein, enzyme is a protein. Everything which is acting like a catalyst, glucose, everything which is acting like a catalyst is a protein. The existence of man, the survival of the man depends upon how the proteins are produced inside the body, which is called as protein synthesis. Abnormal protein synthesis means abnormal hormonal release. Abnormal protein synthesis, abnormal blood or total abnormal functioning of the body will take place. So, if the protein synthesis is proper, then the functioning of the body will be proper. If protein synthesis is abnormal, then the abnormal functioning of the body will happen. So, ribosomes is responsible for protein synthesis. Ribosomes is responsible for protein synthesis. At the time of protein synthesis, tomorrow the topic will come. At that time we will understand how this ribosomes is responsible for protein synthesis. So, one of the major function of ribosomes is protein synthesis. Endoplasmic reticulum. This is called as a bridge like structure. This is called as endoplasmic reticulum. It is a system of membranous tubules and sacs. The major function of endoplasmic reticulum is it helps in the movement of molecules from one part of the another. So, from one part to another part. For example, mitochondria is producing the energy. That energy should go to all the nook and corner of the cell. So, who is pumping this energy to the nook and corner? The waste is produced by every component of the cell. So, who is accumulating this waste? So, endoplasmic reticulum is responsible for movement of the molecules. It might be energy, it might be waste, or sending the food from the cell membrane to the mitochondria. The endoplasmic reticulum will be acting like a transportation medium. Endoplasmic reticulum, which is a tubules attached, it is a tubules which is connected, it is sacs which is connected, is responsible for the movement of the molecules. That is the main duty of endoplasmic reticulum. Then there is nucleus. In between there is nucleus. In this nucleus chromosomes are there. In this nucleus there is chromosomes. Nucleus contains thread-like structures called chromosomes. Thread-like structures which are called as chromosomes. These chromosomes consists of genes. The major function of nucleus is it acts as the control center of the activities of the cells as well as inheritance. Understand here properly. Nucleus. In nucleus, the chromosomes are present. The thread-like structures are called as chromosomes. The thread-like structures are called as chromosomes. If I go in detail, the zigzag, the winded form of chromosome is called as chromatin, and differentiated form of chromosomes which are arranged is called as chromatid, and the element which are present in the nucleus is called as chromosomes. What is this chromosomes? For example, if I take human being, in human being there are 23 pairs of chromosomes. In that 23 pairs of chromosomes, 22 pairs of chromosomes are asexual chromosomes, and 23rd pair is called as sexual chromosomal pair. So, pair means 2, so overall in a human being 46 chromosomes will be there. So, inside the body only 46 chromosomes will not be there. In every cell 46 chromosomes will be there. In every cell 46 chromosomes will be there. So, 46 chromosomes are there in the body means not across the body 1, 2, 3, 4, 46. In every cell there is 46 chromosomes will be there, which is arranged in the form of 23 pairs of chromosomes, which is called as karyotyping, which I will be explaining in 9.1. So, in the nucleus there are 23 pairs of chromosomes. The threaded form of the threaded form structure is called as chromosome, and the winded form of chromosome is called as chromatin. The arranged form of chromosome is called as chromatid. Chromosome is functional, structural and functional part of cell. How? For a human body, cell is structural and functional part. In the same way, for a cell, the chromosome is structural and functional part. The cell is because of chromosome, the cell function is because of chromosome. The directions given by the chromosome is responsible for the proper functioning of cell. So, how the cell is responsible for the man, in the same way the chromosome is responsible for the proper functioning of the cell. So, chromosome is for the cell is for human body. Again, whether it's chromosome or something else. So, this winded form of chromosome, when I open it, when I see it, chromosome is nothing but an arranged format of DNAs, deoxyribonucleic acid. It's an arranged form of DNAs. The DNAs which are arranged in the form of a kilometers, that particular arrangement of DNAs is called as chromosome. Means what? The basic building block of chromosome is what? DNA. The chromosome is because of DNA, but DNA is only structural part. DNA doesn't have any function. Any DNA just it gives information to us. Oh, this is the DNA, that's it. It doesn't have any function. Then what is function? Arrangement of DNAs in a sequence. For example, ATCG. It's an arrangement of DNAs. The arrangement of DNAs like ATCG or CGAT, this arrangement of DNAs is called as gene. Gene is functional unit of the chromosome. DNA is structural unit of chromosome. Understand this. This is chromosome. This chromosome is constructed because of these are DNAs, individual DNAs structures. For example, ATCG, etc. This is structural part of the chromosome. Arrangement. DNAs, when they arrange in the form of a structure, this ATCG will have a function. When the DNAs are arranged in the form of a sequence, a particular sequence, oh, it is ATCG, then it will be having a function like, we cannot understand. DNA, A is a letter, P is a letter, P is a letter, L is a letter, E is a letter, it is an arrangement. But when it have a function, when I arrange it, now it have a meaning, now it have a function. In the same way, the DNA is a structure. DNA is a structure of the chromosome which doesn't have any function. Individual DNA doesn't have any function. Why? Because it's a structure like letters. These letters, when they are arranged properly in a particular format like apple, it have a meaning, it have a function. In the same way, every DNA is a structural part of the chromosome. This structural part of the chromosome, when they are arranged in the form of a determined sequence, the determined sequence is called as gene. So that sequence will have a function, like how apple have a meaning, the DNA sequence will have a function. That functional part of the chromosome is called as, that functional part of the chromosome, this is very, very basic, lifelong basic this is. The structural part of the chromosome is called as DNA. DNA per se doesn't have any function, but the DNAs, when they are arranged in the sequence, then it will have a meaning. Having a meaning is called as function. So the functional arrangement of the DNAs in the chromosome is called as gene. Gene means not object. DNA means object. Chromosome is combination of DNAs. So in that combination of DNAs, if a sequence is having a function, that sequence which is having a function, that particular part is called as gene. So gene is called as functional part, DNA is called as structural part. So the DNAs will arrange in the form of chromosome. Chromosome doesn't have a function, DNA doesn't have the function. On the chromosome, the gene have a, my color depends upon my gene. What is gene? A sequence of DNAs. My eye shape, whether it is oval or round, depends upon the DNA structural arrangement, which is called as gene. My intelligence, my IQ, my words, my sayings, my energy, my physical fitness, my immune system, each and every aspect of my human body is because of the gene. What the information the gene is giving, accordingly my functions will happen. So the gene will give an order to the cell how to function. So generally what we tell, chromosome will be giving the order for the functioning of the cell. In reality it is the gene which is giving the order for the functioning of the chromosomes, are the combinations of the genes. Gene, that the combination is called as chromosome, or DNA combination of DNAs is called as chromosome. Chromosomes are considered as structural and functional units of the cell. Chromosomes carry out all the information that helps the cell to grow, survive and reproduce. The chromosome is responsible for cell to grow, survive and reproduce. Chromosomes. Sir, whether it is really chromosome? Again, when I go deeper, it is not the chromosome. In chromosome, genes are there. So generally we tell that yes, chromosomes are the functional part which is responsible for the survival, reproduction, growth, immune system, everything. When I go into the subject of chromosomes, whether it is chromosome means in chromosomes the DNAs are there, the DNAs are arranged in the form of a structure. That structural arrangement of DNA is responsible for a function. That functional part is called as gene, the structural part is called as DNA. Whole together chromosome is called as structural and DNA, structural and functional part. Why? Because DNAs are present and genes are present inside the chromatid. What is chromatid? See, what is chromatid? Nucleus, winded form, winded form. Cell division is inevitable. For the survival of the man, for the growth of the man, for growth and development of the man, the cell division is inevitable. So cell, how it divides, sir? This cell will not divide. Understand this. This cell will not divide. Inside the cell, then there is a nucleus. Inside the nucleus, there is a chromosomes. Inside the chromosomes, there is a DNA. The DNA starts replicating. Means one DNA will become 2 DNA. 2 DNA will become 4 DNA. 4 DNA become 8 DNA. Means what is happening? The size of the nucleus is increasing. The size of the nucleus is increasing. When the size of the nucleus is increasing, nuclear membrane will be developed. So one nucleus will become 2 nucleus. When 2 nucleus are formed, again cell membrane will be developed. So one cell will become 2 cells. So one cell became 2 cell means cell division. One is becoming 2 means cell division is happening. So the growth of the man depends upon the growth of the chromosomes. So at the time, what is happening? My DNA is getting replicated, doubled. So in the process of replication, what will happen? The winded form of chromosome at the time of cell division, it will arrange itself in the form of chromatid. When it is arranged properly, when anything, if I want to divide, it should be arranged or it should be in a zigzag manner or winded form. it should be arranged. So at the time of cell division, the chromosomes will be arranged in the format, in a specific format, so that the division will become easy. So that arranged format of chromosome is called as chromatid. Cell division at which phase means metaphase, that I will tell in cell division. Cell division at metaphase stage, chromosome will be clearly visible as a symmetrical structure, I told you, symmetrical structure, which is called chromatid. When they are ready for division, they arrange themselves in a symmetrical structure. In a symmetrical structure, when I want to do something, I have to arrange it. When you want to study, you have to arrange the notes. In the same way, when I am going for chromosomal division, it should be arranged properly. That arranged format of chromosomes at the time of cell division in the metaphase stage of mitotic division is called as chromatid. What is chromatin? So these chromosomes which are in the winded form is called as chromatin. Generally it is called as chromatin. It is a mass of genetic material composed of DNAs in a winded form. In the nucleus is chromatin. So inside the nucleus, vital chromosome is present. Inside the nucleus, chromosome is there. When the chromosome is ready to divide, it will be arranging. That arranged format of chromosome is called as chromatin. So inside the nucleus, when the chromosomes are arranged in a winded form, at that time the chromosome is called as chromatin. Arranged format. When you are ready to write an exam, you will be arranging yourself. When you are writing an exam, you will go to examination center and within 2 minutes of exam start, you yourself will be arranged mentally and physically. Chromatid. Generally out of the examination hall, how you will be? Chromatin. Who you are? Chromosome. So you are a chromosome and generally you will be like a chromatin. When you are in arranged format is called as. So these are the cell components. What are the cell components? First is cell membrane, cytoplasm, mitochondria, ribosomes, endoplasmic reticulum, nucleus, chromosomes, chromatid and chromatin. No need to go what they are doing is unnecessary to us. Oh, these are the components of the cell, basic knowledge. So this is what called as chromosome, this is what called as DNA, this is what called as gene. This is what called as chromatid, this is what called as chromatin, this is what called as endoplasmic reticulum, this is what called as mitochondria, this is what called as cell membrane, this is what called as nuclear membrane. The DNA structure will be there. Like this, the DNA structure will be there. DNA structures. Every DNA structure have a nucleotide. One nucleotide will be combined with another, one nucleotide will be combined with another, one nucleotide will be combined with another. So it is called as polynucleotide. It's a combination of nucleotides and the combination of nucleotides is called as polynucleotides. So now we will discuss about DNA structure. This structure. Again I will come back to this figure, you will understand. DNA structure, then DNA replication. DNA structure, DNA replication. DNA, the building block of chromosome. Chromosome, the building block of cell. Cell, the building block of human body. Nucleus contains chromosomes which are visible as rod shaped structure like this. These are rod only. Rod shaped structures. When the cell is about to divide, for example like this it will be looking like. When it is about to divide, so this will be pushed to this side, this will be pushed to this side. Rod like structures at the time of cell division. Chromosomes contain the information. What this chromosomes contains? The chromosomes contains the information of inheritance of traits from parents to next generation in the form of DNA molecules. Means in the division, my father 23 chromosomes and my mother 23 chromosomes combined to form me, which is 46 chromosomes. I am a man of 46 chromosomes and me, I got 46 chromosomes, 23 from my father and 23 from my mother. So in this process, when I am getting 23 from my father, 23 from my mother, so my father will lose 23 chromosomes, my mother will lose 20, no. So that will be divided and they will give my share. It will be divided and they will give my share. So in this way, the division happened and formed in the form of 46 chromosomes. At the time of division of chromosome there, the chromosomes are arranged in the form of a format which is called as chromatid and pushing will take place. So, while I am taking 23 chromosomes from my father, 23 chromosomes from my mother, I will be taking the information which is present in the chromosomes. So, when I am getting something with them, that also will come. When I am taking book from you, only book will not come. In the book pages, what is written also I will get it. If you have a diary, you are writing daily diary, and I took your diary, means not only diary will come, I will know the information which is present in the diary also. So, when I am taking the 23 chromosomes here, 23 chromosomes here, not only the chromosomes, with the chromosomes what I am getting? The information of my father, the information of my mother, both combined into me to form 46 chromosomes. So, in me, whose information is there? My father's information is there, my mother's information is there. So, what is this information? Inheritance of trait. It is called as inheritance of trait. It can be a biological inheritance, it can be intellectual inheritance, it can be immune system inheritance. What information is present in the DNA? That 50% in me is my father's information, 50% from my mother's information is present in me. So, that information can be related to protein synthesis, that information can be related to disease, etc. So, me is filled with information of my father, me, I am filled with information of my mother. So, I am not unique, I am not a separate entity. In me, whatever information is there, it is pumped from my father and mother. So, that pumping of information from my father and mother equally is called as inheritance of. So, chromosomes I got. It is not the chromosomes. In the chromosomes what are there? DNAs. Why? Because DNA A is one information, T is another information, C is another information, G is another information. The combination of A, T, C, G is a function. So, that is imbibed into me. In the same way, for my mother also imbibed into me. So, in that way, inheritance of traits are happening inside. The functional segments of DNA are called as. So, structural segment of chromosome is DNA. DNA is just a structure like apple. A is a structure, P is a structure, P is a structure, L is a structure, E is a structure. The combination of APP, LE is a function. Combination of APP, LE is a function. So, that functional element is gene. The structural element is DNA. Where? Of chromosome. DNA molecules contain the information for constructing and organizing the cell. So, that DNA will tell A, T. Accordingly, the cell will be formed, the tissue will be formed, the organ will be formed, and the human body will be formed. So, the cell is formed because of the information which is coming from the DNA, and the DNA is giving the information how the human body should function. DNA is the information source for making proteins. If the information is altered, I will tell you, very, very important. In my body, it might be manufacturing of hormones. It might be manufacturing of enzymes, manufacturing of lipids, manufacturing of any catalyst in my body is because of protein synthesis. It's nothing but production of proteins, is production of these things. In protein synthesis, what happened? From the DNA level, we will take the information and we will produce a hormone. This is general format. In protein synthesis, we will take the, tomorrow protein synthesis will come. I will take the information from DNA and I will produce protein. So, this produced protein is because of what? The information taken from DNA. So, this protein is because of what? DNA. So, what information is present in the DNA is responsible for the formation of protein. DNA is the information source for making proteins. If the information is altered, if the information is wrong, then protein will be wrong. If the information is wrong, then the protein will be wrong. I took the dairy. With the dairy, what I got? I got the information also. If the information is wrong, what I am interpreting will be wrong. So, if the information is wrong, then obviously the function will be wrong. If the information is altered, then unique proteins are formed which will eventually lead to change in the body functioning. When the information is wrong, suddenly nuclear radiation came. Suddenly chemical agents, I got exposed to chemical agents. I got exposed to nuclear radiation. For example, in Kaiga nuclear power plant of Karnataka. Kaiga nuclear power plant of Karnataka, recently what happened in 2016, the drinking water fridge got polluted with nuclear radiations, with some nuclear elements accidentally. A high level inquiry committee was being constituted, and the scientists in the Kaiga nuclear power plant got impacted with some genetical diseases. So when they did inquiry, why they suddenly got some genetical abnormalities, genetical diseases, the inquiry committee has constituted and told that it is because of a fridge got impure or got polluted because of some nuclear elements. So the people who consumed that water, they got their DNAs got altered, because nuclear energy have huge power. It have the power to mutate the DNAs, mutate the gene, mutate the chromosome. So what will happen when this mutated DNA will be responsible for abnormal chromosome, abnormal hormone or not, abnormal protein synthesis or not. So when there is wrong information, the protein produced will be wrong. If there is alteration in the information in the DNA, then there will be alteration in the production of protein. If protein synthesis is wrong, the man will die. If protein synthesis is wrong, the man abnormality will be there. If protein synthesis is wrong, the man will have some or the other diseases. So the information which is coming from DNA is responsible for the functioning of human form, which will eventually lead to change in the body functioning. So what is DNA structure? DNA has three main components. As I told you earlier, DNA has three main components, which together is called as nucleotide, This have three things. One nucleotide have three things. The combination of nucleotide is called as DNA. Deoxyribose, which is called as pentose sugar molecule. Base, which is called as nitrogenous base. There are four different bases: adenine, thymine, guanine, cytosine. There are four different nitrogenous bases. Phosphate or phosphoric acid. So every nucleotide is a combination, or DNA has three components, which is called as deoxyribose, nitrogenous base, phosphate. These three combines to form a DNA, or to be precise, these three combine to form a nucleotide. The combination of nucleotide is called as a DNA. DNA has three components: deoxyribose, a pentose sugar molecule, deoxyribose, a pentose 1, 2, 3, 4, 5. Pentose means 5. So 5 pentose sugar molecule, which is connected to nitrogenous bases. This is nitrogenous base. Nitrogenous base connected to a pentose sugar molecule to a phosphate. This is a combination. These three together is called as nucleotide. These three together is called as a nucleotide. This will join again another nucleotide. So the combination of nucleotide is called as DNA. So one DNA is one nucleotide, and the combination of nucleotides is called as DNA, which is one nucleotide is nothing but a combination of pentose sugar, nitrogenous base and phosphate, one by one. First nitrogenous bases, this one, deoxyribose, pentose sugar, this one, nitrogenous bases. There are four different bases are there: adenine, guanine, thymine and cytosine. The bases, the nitrogenous bases. So we are understanding the we are understanding the cell. From cell we came to nucleus. From nucleus we came to chromosome. From chromosome we came down to DNA. In DNA we are understanding what are the structures of DNA. So every DNA is a combination of a nucleotides. One nucleotide is nothing but a pentose sugar molecule and a nitrogenous base as well as a phosphate. So first about nitrogenous bases. Nitrogenous bases are four, which are divided into two: A, T, C, G is divided into two, which are named as pyrimidines, purines. It is divided into two, pyrimidines and purines, based upon the structure. In your school organic chemistry you studied COH structures, or based upon the structures it is classified as pyrimidines and purines. What are pyrimidines? Made up of 6-member ring: 1, 2, 3, 4, 5, 6. If 6-member single ring is there, it is called as pyrimidine. Thymine and cytosine. thymine and cytosine. Pyrimidine. Purine made up of 6-member ring fused to another member. 1, 2, 3, 4, 5, 6 fused to another member. It is called as purines. A and G. A and G. Adenine and guanine. Adenine and guanine. So there are pyrimidines and purines. There are pyrimidines and purines. So pyrimidines are thymine and cytosine. CT, remember it like CT. CT, computer scan, CT scan. AG, Attorney General. AG is purine, Attorney General is pure. AG. Pyrimidine, CT scan. Because of CT scan, the prices is increasing like pyramid. Pyrimidine, CT, cytosine and thymine. Whereas purines are Attorney General. The ring are made up of, these rings are there, no? They are made up of carbon and hydrogen. Like this it will be, structure. COH. Arranged structures will be there. Structures will be there. No need to understand that. So 6-member ring is called as pyrimidine. 6-member structure which is fused to another structure, another ring is called as purine. Pyrimidines are CT, cytosine and thymine. Purines are AG, adenine and guanine. One pyrimidine will match with another purine. We will look into it later. So first you understand nitrogenous bases, pyrimidines and purines. What is this? Phosphate. What is this? Pentose sugar molecule. What is this? Nitrogenous base. This combination is called as nucleotide. The combination of nucleotide is called as 1-DNA. Nucleotides are formed by the condensation of a pentose sugar, phosphate, pentose sugar and one of the 4 bases. It can be A, it can be T, it can be C, it can be G. So if it is one nucleotide. In this way, 3 nucleotide combine to form a DNA. 3 nucleotide combine to form a DNA which is called as polynucleotide. More than one is called as poly. So polynucleotide is DNA. So 3 structures combine to form a DNA. What is that? It is a combination of phosphate, pentose sugar molecule and nitrogenous base. Nucleotides are linked together by covalent bonds called phosphodigester linkage like this. In this way the link will be happening. In this way the link will be going. DNA. One is called as nucleotide, another is called as another nucleotide. So one nucleotide is joined with another nucleotide is joined with another nucleotide. By what? Phosphodigester linkage. A covalent bond, each will be associated with other. It is phosphate, it is pentose sugar molecule, it is called as nitrogenous bases. The nitrogenous bases is divided into pyrimidines and purines. Pyrimidines is CT scan, cytosine and thymine, and purines is Attorney General, adenine and guanine. DNA double helix hydrogen. This is one nucleotide, another nucleotide. So this nucleotide will join with another nucleotide. This nucleotide will join with another nucleotide. This nucleotide will join with another nucleotide. Double helix structure. 2 are there. Double helix structure which are anti-parallel. Means if I open it, it will not be like this. Same, it will not be like this. If I open it, it will be 5 dash, 3 dash, 3 dash, 5 dash, which are anti-parallel. Means the end point of here will join here with a cut in between. So these are anti-parallel, not parallel. Made of 2 strands of nucleotides, yes or no? 2 strands of nucleotides that are joined together by hydrogen bonding. The 2 are joined by one line is there in between. That line is called as hydrogen bonding. Hydrogen bonding occurs as a result of complementary base pair. Here, this nucleotide of one strand and the nucleotide of another strand will join together with a bond called as hydrogen bond. So how this joining will be happening? One pyrimidine will join with another purine, one pyrimidine will join with another pyrimidine. How, sir? A with T, C with G. Pyrimidines are CT, purines are AG. So A with T, C with G. A with T, C with G. A of purine is associated with pyrimidine of T. C of pyrimidine is associated with G of purine. One base pair, one pyrimidine will associate with another purine. One pyrimidine will associate with another purine. CT, C will be associating with G and A will be associating with T. It's fixed formula. When this link is established, hydrogen bond will be there. This combination of nucleotides is called as DNA. Adenine and thymine pair up, cytosine and guanine pair up. Adenine and thymine will pair up, cytosine and guanine will pair up. Each pair is connected through hydrogen bond. This line is what? Hydrogen. A connector between the two nucleotides. A connector between the pyrimidine and purine is what? Hydrogen bond. Hydrogen bonding always occurs between one pyrimidine and one purine. C, G. Between C and G, 3 hydrogen bonds will be there. Between A and 2, there will be 2 hydrogen bonds will be there. So what is this lines are in between? This is hydrogen bond. This is hydrogen bond. When C with G, when A with T, when C with G, when A with T, it is the DNA structure. One nucleotide in combination with another nucleotide, one nucleotide in combination with another nucleotide, one nucleotide combination with another nucleotide. The 3 nucleotides combination is one DNA generally. What are pyrimidines? CT scan, pyrimidines. Cytosine, thymine. What are purines? Attorney general. Adenine and guanine. So one pyrimidine will match with another purine. Means one A with another T. One C with another G. So if it is between C and G, it is triple bond. Between A and T, it is double hydrogen bond. Double helix and hydrogen bonding. C. Guanine, cytosine, adenine, 2 hydrogen bonds. This is DNA structure. In this way they are. This is inside. Generally, this is what? These lines are these bonds. These lines are these bonds. So, how it got joined? One pyrimidine, one purine. One pyrimidine, one purine. One pyrimidine, one purine. So, this joining is called as hydrogen bond. The 2 strands 5' 3' and 3' 5'. In between the 2 strands which are not same, which are not complementary, which are different. DNA double helix and hydrogen bonding. Adenine always pairs with thymine because they form 2 hydrogen bond with each other. Cytosine always pair with guanine because they form 3 hydrogen bonds with each other. So there is, when I am talking about A and T, 2 hydrogen bond. When I am talking about C and G, 3 hydrogen bond. Genes. In this way, the DNAs are there. So again I am repeating. DNA. When I talk about a single DNA, it's a double helix structure. In double helix structure, on every strand there is a nucleotide. That side, this side, a nucleotide strand will be there. So nucleotide strands will be joined together by hydrogen bonds. The base pairing will happen with one purine with another pyrimidine, one pyrimidine with another purine. Like A with T and C with G. So this combination of 2 nucleotides on 2 strands, the combination is called as DNA. This DNAs, when they are arranged in the form of a structure which is having a function, is called as this DNAs, when they are arranged in the form of a structure and that structure is having a function like A, T, C, G. Here A, T, C, G. A for T, T for A, C for G, G for C. That combination, that arrangement is called as gene, which is having a function. Genes, units of genetic information, hereditary information. Order of nucleotides make up genetic code. Genes can contain the information for one polypeptide, one nucleotide combination is called as polypeptide. That polypeptide together is a combination of DNAs. Combination of DNAs, arrangement of GNAs is called as gene. Genes can also regulate how other genes are expressed. All cells of an organism contain the same genetic information, but they do not all express the same genes. Means on the chromosomes, on the genes, there will be expression for every gene. There will be expression for every gene. The gene, the structure is ATCG on chromosome number 21 and the structure ATCG on the chromosome number 22 have different functions. The information might be same, but according to the chromosome number, the function will be. The apple might be the same, but apple in English language and shape in another language. So when it is present in another chromosome, the same will be by different function. The same will be by different function. So the informations will be same. Why? Because I am taking from father and mother is same. So the information in my all genes will be same, but its function will be differing in which chromosome it is. Or simple, will not go deeper. So genes have a function. So that gene which is having information, if it belongs to chromosome number 1, one function. If it belongs to chromosome number 2, one function. If it belongs to chromosome number 3, one function. So based upon the chromosomes, the functions will be there. For example, chromosome number 12 is responsible for, in chromosome number 12, a part of chromosome which is a gene is responsible for melanin production. Melanin is a protein. Melanin production. So the same structure of gene is present in other chromosomes also, but that structure of gene which is present in the chromosome number 12 is responsible for my color, which is nothing but melanin production. Or my melanin is because of that gene which is present on the chromosome number 12. The same information if it is present on the chromosome number 13, the function will be. Means what? These genes are having functions. General understanding is every gene have a function. Again, I am summarizing now as an answer. If there is a question on DNA structure, DNA is a double strand, 2 strands, helical structure, like this helical structure. DNA has 2 polypeptide nucleotides, 1 polypeptide, this is polypeptide nucleotide strand, polypeptide nucleotide strand. Polypeptide nucleotide consists of 3 components. Every nucleotide strand is a combination of phosphate, pentose sugar and nitrogenous base. And in nitrogenous base there are purines and pyrimidines. Purines are adenosine, adenine and guanine, and pyrimidines are cytosine and thymine. Ratio of 1 purine and 1 pyrimidine. 1 purine will match with another pyrimidine, 1 pyrimidine will match with another purine. So the ratio of purines and pyrimidines will be 1:1. Then only proper pairing will take place. When there is proper pairing, it is called as DNA. If improper pairing, DNA abnormality. DNA abnormality, gene abnormality. Gene abnormality, chromosomal abnormality. Chromosomal abnormality, cell abnormality. Cell abnormality, tissue abnormality, organism. Every purine will match with another pyrimidine. A with T, adenine matches with thymine through double hydrogen bond. Cytosine matches with guanine with triple hydrogen bond. This nucleotide contains 2 DNA strands. These nitrogen bases carry the information which are responsible for inheritance. DNA strands are anti-parallel to each other, 5', 3', 3', 5'. Pentose sugar, this component holds phosphate and nitrogenous base together. Pentose sugar has 5 carbon molecules. Whereas phosphate, what is the function of this one? I am talking about this pentose sugar molecule. So what is this pentose sugar? It will carry the nitrogenous base, it will carry phosphate. It is a energy provider. It will carry this both. And this will go into the next. This is chain link is because of this pentose sugar molecule. Phosphate, also called as phosphoric acid, is responsible for the formation of nucleic acid. All these 3 components together constitute a DNA strand. It holds the strand. The phosphate holds the strand. So the support is given by phosphate, and the pentose sugar molecule associates with nitrogenous base. The main part is nitrogenous base. Functions of DNA, it reveals the secret of protein synthesis. Why? Because the information which is present in the DNA is responsible for the production of protein. So the protein synthesis is mainly because of DNA. The operation or functioning of a gene depends upon the structure of DNA. APPLE. APPLE is DNA. If one P is absent, whether gene is wrong or not, if one P is absent, so the spelling will be correct or not, the pronunciation will be improper. Why? Because one P is absent. So the structure of DNA is responsible for the functioning of gene. If there is abnormality structure in DNA, then there will be abnormality in gene. General understanding of DNA. Online, who will explain me back? Complete DNA structure. Raise your hand. So this is about DNA structure. Again, I will revise. I explained about cell, how cell got discovered, then cell components, various components, cytoplasm, mitochondria, ribosomes, endoplasmic reticulum, chromosomes, chromatid, chromatin. This is important for our 9th unit. This is our syllabus, so I focused here more. So what is this DNA structure? We understood. What is gene? We understood. What is chromosome? We understood. DNA structure. We have, it's a combination. A nucleotide is a combination of deoxyribose, nitrogenous base and phosphate. In this way the structure is there. So there are pyrimidines and purines, one will match with another. So through hydrogen bonds 2 and 3, the combination of nucleotides is nucleotide strands which are called as polynucleotide. One polynucleotide have another matching with another polynucleotide strand which is called as strands, which are anti-parallel to each other. So one will be matching with another. If there is abnormality in DNA, then there will be abnormality in gene level. Then this is summary of the DNA structure. This one topic we will discuss and we will take break. Tomorrow, day after tomorrow. Tomorrow there is no class. Tomorrow you have test number 7. Complete socio-culture anthropology question paper. Complete socio-culture anthropology 2, 3, 4, 5 on UPSC standard it will be there. Okay. It will be of the level of test series. DNA replication. Just to remember, now growth and development of me should happen. I should have growth and development. A baby should grow and develop. So how the growth and development will happen? How your growth and development will happen? So growth and development is nothing but multiplication of cells. If more the cells, more the size. If the cells is more the size. More the size means growth. If proper growth, it is called as development. Just growth, it is not called as development. We have some people, growth will be there, but there will be no development. So growth means only cells, development of cells. How it will happen? So cell have nucleus. In nucleus there is chromosomes. So chromosomes need to be doubled. When the chromosomes are doubled, then the nucleus will be doubled. When the nucleus are doubled, then the cell will be doubled. When the cell is doubled, the size will increase. So that means the chromosomes should be doubled. What is the structural element of chromosomes? DNA. DNA should be doubled. Doubled means replication, copy. Replication means copy. It is copying. So 1 is there, 2 is. 2 is there, 4. 4 is there, 8. Doubling is happening. Replication is happening. Copying is happening. When the doubling is happening, the DNAs are increasing. When the DNAs are increasing, then the chromosomes will increase. When the chromosomes are increased, then the nucleus will increase. So that means for the growth and development, replication of DNA is important. Doubling of the DNA is important. So how this doubling of the DNA will happen? In the DNA replication, the major role is of enzymes. Enzymes will act as catalysts. Enzymes will act as catalyst. If there is no enzyme, then the DNA replication will not happen. So the doubling of the chromosomes is happening because of the doubling of the DNA. The doubling of the DNA is happening mainly because of the role of enzymes. Enzymes have a huge role, like catalysts, like chemicals. When we use chemicals, sudden burn will happen. So it is the sudden burn is happening because of the chemicals. So these chemicals are catalysts, which are named as enzymes. So major role is enzymes. You have to remember the enzyme and what it is doing. You have to remember the enzyme, what it is doing. 6 steps, step by step we will be understanding. 6 steps are there. 6th step. 6 steps in DNA replication. First step, second step, you can also write in flowchart format. DNA replication. DNA replication can be observed at the phase of interphase during the division of cell. I will tell you. See here. This is cell division. This is cell division. In this we have M phase where the division will happen. This is mitotic phase, means this is mitotic phase. Before mitotic phase, after mitotic phase, this is called as interphase. This phase is called as interphase. In this what happens? Here only division will happen. So before division the number should increase, no. Before the nucleus division happens, cell division happens, first chromosome should be divided. So that chromosomes division happened in this S phase. So in the division of chromosomes, what replication should happen? DNA replication is happening. So where the DNA replication is happening? Interphase or S phase, which is called as synthesis phase. What is this? Cell division cycle, which I will be explaining tomorrow. Cell division. In this cell division phase, except mitotic phase, it is called as synthesis phase. In this synthesis phase, the DNA will be doubled. Because the DNA is doubled, the chromosomes are doubled. When the chromosomes are doubled, then the cell division will happen. So the division of cell will happen in mitotic phase. Before the cell division, what all is happening is this 95% of the time, which is called as S phase or synthesis phase. The same thing is here. DNA replication can be observed at S phase, synthesis phase of interphase during the division of cell. In that complete cell division process, except mitotic phase, it is called a synthesis phase. In the mitotic phase, the cell division will happen. So before cell division, what should happen? The DNA should increase its size, the DNA replication should happen, the chromosome replication should happen, then the cell division will happen. So this DNA replication is happening where? In the synthesis phase. So in this synthesis phase, there are 6 steps. Number 1, winded form of chromosome. Anything which is not uniform, anything which is disorganized, we cannot teach them a lesson or we cannot divide them. So first duty what I have to do? First I have to organize the chromosomes. So these chromosomes, I started organizing them. Means what? Winded form of chromosomes, I made it unwinded form. I opened it. When I made in unwinded form, it got arranged. When it got arranged, I can easily divide it. The first step is unwinded chromosome became winded. Winded chromosome became unwinded. Unwinding of DNA takes place, chromosome within the chromosome DNAs, by an enzyme called as helicase. First enzyme. Helicase. This is the first enzyme. The first enzyme helicase is responsible for unwinding of the chromosome, which is winded. I made it into unwinded. I means I arranged it. Second step. Initiation. Now replication, copying should happen. Copying should happen. So this I opened it, which is winded. I unwinded it. So it became line parallel. It was unwinded, it was winded, so I made it into unwinded. So now it got differentiated, it got separate lines. How it got winded into unwinded? By an enzyme called as helicase. Now it got properly divided, properly got separated. Then one more enzyme which is called as primase. Initiation is starting. Primase. My target is what? This should be double. One line is there, another line is there. Means 2 are there, should become 4. Means doubling. Replication means what information is present here should be replicated here. What information is present here should be replicated here. So 2 strands became 4 strands, doubled, replicated. But DNA has one disadvantage. DNA itself cannot generate another strand. So what the primase enzyme Whereas RNA is single strand or double strand with your 9th class knowledge? Single strand. So one RNA will come here. The primase enzyme will release one RNA. So it will initiate up to here. Primase will release one RNA primer. Why? Because the initiation will not happen with DNA. Why? Because DNA is a double strand. So one RNA primer will be released. Why? Because a single strand it will come here because of an enzyme called as primase. Now here the base pairing happens. Only one difference, when here it is T, there will be U. In place of thymine, for RNA there will be uracil. So when RNA primer has established up to here, so one foundation has been formed. One foundation has been formed. Now here the RNA primer will stop and DNA polymerase, third enzyme will come, and this continuation to RNA primer, already I got the stability, so a DNA will be produced here as a continuation to RNA primer. So this DNA polymerase will be matching this DNA with this DNA, with this DNA with this DNA. We'll come back. Primase is an enzyme released from an RNA primer in order to initiate DNA duplication. The initial impetus for DNA replication is given by RNA primer because RNA is a single strand. So RNA is single strand, so it can come. DNA cannot come. Why? Because it is a double stranded. But I required only one strand. When I required only one strand, so the primase enzyme, a RNA primer came, the RNA primer started initiation. That is the reason the second step name is called as initiation. When the initiation happened, so then I stopped RNA primer, then a DNA polymerase. Formation of DNA strands, daughter strands begins with the help of DNA polymerase. So continuation to it, here the RNA primer will stop and this RNA primer will be removed and the DNA daughter strand will be formed because already we got the support of RNA primer. It is continued by DNA strand. And this DNA daughter strand will match with the parent DNA strand, will match with the parent DNA strand. Same thing will happen this side. A RNA strand will come, initiation will start. When initiation started, a base has been formed, then DNA polymerase will be responsible for the formation of daughter DNA strand. This daughter DNA strand will match with the parent DNA strand. So parent DNA strand, a daughter DNA strand, a daughter DNA strand, a parent DNA strand. In this way, one strand has become two strands. One strand, this which is one strand became two strands. Formation of DNA strands. Daughter strands begins with the help of which enzyme? DNA polymerase. So first enzyme is helicase, which is unwinding the chromosome. Second, when it got unwinded, I need to produce the copy. When I want to produce the copy, DNA cannot initiate itself. Why? Because DNA is a double strand. I want only a single strand. I want only a single line. So a RNA primer will be coming because of an enzyme called as primase, and to the DNA strand a primase will be responsible for the formation of RNA primer. So the DNA will be matching with RNA primer. But once the initiation is happened, the RNA primer will stop, DNA polymerase will be responsible for continuation of RNA primer into a DNA primer. Why? Because the stability is given by the RNA primer. So when this RNA primer, then the RNA primer is removed and the DNA strand which got continuation of RNA primer, because of DNA polymerase strand, a parent DNA and a daughter DNA strands both will be evolving. So this side also same, this side also same. So this strand became one, this strand became one. So one is helicase, other is primase, other is DNA polymerase. Exonucleus enzyme, RNA should be removed, no? Helps in removal of RNA primer. Eventually DNA polymerase helps in stabilizing the complete formation of DNA strand. That RNA primer is removed by which enzyme? That RNA which is helped for DNA polymerase to form the continuity of a DNA, but I don't want DNA strand. So DNA strand or DNA primer is removed by which enzyme? Exonucleolus. So exonucleolus enzyme will be removing this DNA, for example. Child when he unable to talk to any stranger, then the mother or father will come and introduce, then he will go to the stage and he will speak. In the same way, the DNA itself cannot be formed into another copy. So, RNA primer, which is a single strand, it will come like an RNA primer and starts initiation. After initiation, when it both got, so double, so already one support is there, to continuation to it, a DNA polymerase enzyme will be releasing a DNA strand. So, to the RNA primer, the DNA strand will be continuity. So, I no need now RNA primer. So, RNA primer should be removed. This RNA primer will be removed by which enzyme? Exonucleolus. So, this remaining DNA will increase its length. So, RNA primer is removed. So, again the base pairing will start with the parent DNA strand and the base pairing happen with daughter DNA strand. It grows up. It grows up. So, helicase, primase, third one DNA polymerase, fourth one exonucleolus. Now, DNA strand, parent DNA strand and daughter DNA strands both got formed. I have to join them by triple hydrogen bond or double hydrogen bond. Matching should happen. Then only a complete DNA is completed. So, this joining of the two strands, parent DNA strand and daughter DNA strand, is happening because of which enzyme? Ligase enzyme. Ligase enzyme is used in joining of DNA segments or DNA strands. One is parent DNA strand, other is daughter DNA strand. Now, see how many enzymes? Enzymes are catalysts. They produce lot of side effects. Why? Because these are externally coming. So, obviously the DNA will become volatile. There will be high pressure on DNA. The enzyme topoisomerase helps in relieving the stress and tension formed on the DNA strand because of, like, so too many tablets will, when we take, there will be tension and stress on us or not. In the same way, when too many enzymes are acting on this parent DNA strand and daughter DNA strand, there will be pressure on the strand. So, there is a chance of collapse of the DNA strand. So, a neutral thing will come which is called as topoisomerase. This topoisomerase enzyme will neutralize the DNA strands. Each DNA molecule contains one parental strand. At the end, DNA rewinding happens. So, each DNA strand will have one parental daughter, parental daughter, parental daughter. 1 became 2, 2 became 4, 4 became 8. In that way, the DNA has got replicated. So, the major role is enzymes. First is helicase, second is primase, third is DNA polymerase, fourth is exonucleolus, fifth is ligase, sixth is topoisomerase. So, in DNA replication, the higher role is of enzymes. So, a time bond, these enzymes will be released. Sir, what is enzyme? Enzyme is a protein. If ligase is improper, topoisomerase is improper, if helicase is improper, primase is improper, DNA polymerase enzyme is improper, because of improper protein synthesis, complete DNA replication will be improper. Enzymes are nothing but proteins. So, in this way, for every aspect of human body, the proteins are responsible. Why? Because enzymes are proteins, hormones are proteins, enzymes, in this topic, enzymes are responsible for DNA replication, enzymes are nothing but proteins. So, what is the best role of protein synthesis is DNA replication. Means doubling of the cell, that means growth and development of the human being is because of enzymes. Enzymes are nothing but proteins. What are the enzymes? Number 1, helicase, primase, DNA polymerase, exonucleolus, ligase, topoisomerase. This is about, if I tell more, you will get confused. So, today you read it. Try to spend time on it. Watch the recorded video, don't open any YouTube video. Just open it, wait 1 minute and think, Acha, yes, when DNA strand is there, it's a double strand. So, it got separated, it became single strand. So, see, DNA is never a single strand. So, DNA is never a single strand, it cannot develop by its own. Why? Because single strand. I want only single strand. Why? Because already parent is there. But RNA is a single strand. So, RNA came. as RNA came, a support system got developed. I continued with it with a DNA strand. So, this DNA strand, another DNA strand, parent DNA strand and daughter DNA strand became DNA. Who helped it? A enzyme is helping it. Enzyme is responsible for this particular process. This is about DNA replication. Complete functioning of the human body, DNA replication, DNA structure, everything is because of this one, protein synthesis. Enzymes are proteins, hormones are proteins, means if this protein synthesis is wrong, enzymes will be wrong. If enzymes will be wrong, DNA replication will be wrong. If DNA replication is wrong, I will die. Why? Because no growth and development. So, how this protein synthesis will happen, we will discuss tomorrow. With that protein synthesis, we will discuss about cell division, mitosis, what is mitosis and what is meiosis, we will be discussing about it. Mitosis, meiosis is easy. Protein synthesis you have to understand. So, today we understood cell, in cell nucleus, in nucleus chromosome, in chromosome DNAs, the structures of DNAs is called as genes, genes are functions, DNAs are structures, 23 from father, 23 from mother, basics, then DNA structure, DNA replication, basics only we discussed. Tomorrow we will, Thursday, we will try to complete protein synthesis and cell division. Still it is basics only. We cannot expect, but still you got married and your 8th class son or 9th class daughter is asking, sir, what is this, daddy, what is this cell division, cell division. Once upon a time cell division, 2 will become 4, 4 will, how means it will become, that is it. All the best for tomorrow's test, test number 7. Thank you.